So you already seen this picture. I'm slightly modified it. Just different between different subjects. Yeah. And this is the first grade.
What is this formula for pi?
Yeah, this is a famous formula for Ramanujan.
It's Ramanujan.
Yeah, it's one of the famous formulas. It's kind of very, very non-trivial formula. Yeah. Yes. But this is how numbers are being mixed with geometry. It's kind of a remarkable formula. And so just I'm repeating. And this is kind of how you describe things in physics. We cannot describe it in words, but we have to only in numbers. You have to describe some probability distribution. This is the surface of Mars, and actually almost everything except for life looks like that. And so our visual system, because sometimes people say you analyze images, but images in life and images not in life, absolutely nothing, almost nothing in common. Right? There is very little common in these two pictures, and the analysis mathematically or with your brain will be different fundamentally, and our perception actually are very adapted to that. We can tell a story about that. We cannot say much about that. We only can do it in numbers, and this is why mathematics, more like biology, this of course is a formula, but it can be described in words if you go deeper into the structure. Some particular fields involved, and well, not that I know that, but you can say it in words. But physics are not expressible in words; it's by nature numerical. And so the specific of life is just very well articulated by Francois Jacob. That is a combination of physics or chemistry plus information somehow in there. So, but I was speaking, the last I was finishing, and I have to say a couple of words about something physical before so the essence from our point of view is informational. And it's very difficult to talk about this in a rigorous term, yeah? But I still want to continue what we're speaking about cells on the physical side. So this is an essential thing to have in mind, the scales of what happens in there, and in one particular instance, kind of scales I think are kind of crucial to understand the relations of different time scales, and this is specifically speaking about enzymes. So first, of course, the size of the cells and what happens in the cell. So cell is reasonably small. And as I was explaining, it is very crucial. It's volume-wise small. On the linear scale, it's 1 micron, 1 millionth of a meter. Of a bacterial cell, about 10 times bigger for our cells. And volume-wise, so our cells 1,000 times bigger. And the content of a cell, the number of atoms or small molecules, like water molecules, in the cell is by order of magnitude or smaller the number of bacteria in a colony of bacteria. A small colony of bacteria, like in your gases anywhere, contains quadrillions of bacteria, and the number of atoms of bacteria of the order of trillion. So it's a really big difference, and therefore the number of configurations of atoms making bacteria is smaller than the potential number of exponentially growing community of bacteria. Therefore, life, if you know the life exists, you know it would exist with probability 1, provided that you live on a time scale, of course, much bigger than the scale of the time of the life of the universe. Yeah? If you imagine you have infinite time, infinite meaning of the order of 2 to the power 1,000, then life will be always there. If just because you know it exists, such a small machinery as bacteria able to reproduce exponentially, right? So this is a miracle. Granted, this miracle, which we certainly cannot rationally explain, we only can observe it and justify it. But granted that, mathematically, it's plausible. And this wouldn't be, and maybe it's not accidental, this balance of numbers, but definitely if it were already like bigger bacteria, amoeba or subject, it would be questionable, because both the number of atoms goes up by order of 1,000, and the number of population accordingly goes down. So But now...
Any other examples which exponentially grow but not bacteria, but I don't know.
No, they grow exponentially, but not with such a rate as bacteria, of course. It was a bigger cell that can't occupy as much space as bacteria. But of course, small eukaryotes, there's still a lot of them. But, well, anyway, as you know, historically, bacteria existed on Earth half of the lifetime of life on Earth. There were nothing but only bacteria, only prokaryotic or these very small cells. And eukaryotes appeared about 2 billion years after appearance of life, right? And then again, for half a billion years, nothing was happening, but still things started developing much faster when two cells merge and you have more complicated cells. But bacteria, of course, were and will be forever, because we can expect dominant constituents, members of participants in life on Earth. But now I want to say a couple of words about enzymes, because here there are some physicists that can help me with that. So there are three basic, on the molecular level, two in their combination, two processes happening in cells. And they related to the fact that there are two energy scales. So one is size scale, then there is energy scale. And the energy, the unit of energy in life is the energy of moving molecules of water or anybody at the temperature we live at 20 centigrade. Yeah, and this is standard units of energy. It's about well, I keep forgetting. I think it's 2 kilocalories per mole, or whatever it is. Yeah, but this is units, and you don't have to know anything about anything else. That's the unit of energy. And then there are two energy levels in these units, measured in these units. One is a weak interaction between molecules. They're a little bit stuck to each other, and the major, and this is essentially because molecules slightly charge, and the charge is not symmetric, so they have dipole attraction, and so there is little attraction between molecules. And the energy of this this is called kT units, and this several kT from 2 to maybe to 10. 10 is a lot, yeah, maybe 6, yeah. So it's comparable to the energy of motion. But the time of they live together, the attraction exponentially goes with energy. So you change energy 2 or 3 times, but affinity goes exponentially in 2 or in 4 times more. So already for small weak interaction, affinity may be very strong. May stick and stay together very long. But still it is by far weaker than even the weakest intermolecular interaction. These covalent bonds, chemical bonds, have much bigger energy, which may be 100 times more, right? So it will be 100 times more, say, for the energy of a green photon, and which already can destroy many of covalent bonds. You know, under the light destroys colors, say pigments in your hair. And so there are these two different energies, and there are accordingly 2 different processes in the cells. One related to these weak bonds, and those related to the stronger bonds. And the weaker, it's physics, they come together, and this is more or less well approximated by classical mechanics, classical physics, and covalent bonds, everything is quantum. And this is so I understand it much less. However, it described, yet description of that usually doesn't explicitly involve quantum mechanical considerations. In any case, we can say about that, but you can't make any computations. But then the 3 basic processes, I said, one with a weak bond, and the major process is self-assembly. When you have some big molecules, combination of molecules, and they have some distribution of this charge, so that you know how they tend to stick to each other. You know it in principle, of course, in practice you don't know. Then they become stuck to each other, and the main process there is protein folding, right? So you have a long molecule of 20, built of basic 20 units, polypeptide chain, and then under nice conditions folds. It's getting stuck, and presumably it takes the minimal energy. So the sticking can minimize the energy of this interaction. This is assumption, which of course very hard to verify. It may be not a minimum, and sometimes not minimum, but kind of it's maybe not quite stable, quite stable, and then there are kind of many other processes of the same type. Once you assemble this molecule, and then it having this kind of self-assembly. There is one kind of self-assembly which is very special, which is, this is kind of very remarkable. It will not just any kind of assembly. If you have to assemble in a very intelligent way, and then take particular form. And that for protein, it's a crucial moment when information turns into really something physical, right? The information written formally in DNA, it's actually description in words, and these words are really words. They have no physical kind of, nothing physical about them, yeah? It's not specifically physical process, physical description. It's symbolic description, and then it materializes, and the moment it materializes, exactly the moment when protein folds. Because there are two other processes, there is something written in DNA, then it's rewritten in a kind of letter by letter to RNA. It's just for certain, there is good reason for do that, why I do it in this step. And then from RNA, you read it now not letter by letter, but 3 letters divided in blocks by 3 letters. And then you have polypeptide chains. And the second process, chemically, extremely complicated. In a second, I say what's involved. But the last is, technically nothing happened there. You just throw it in the water and it falls like that, just because it's actually the law of this molecular interaction of these molecules. So it's easy to say, but again, computationally, whatever, it's completely incomprehensible. And we don't understand why and how they fall. However, now there are beautiful algorithms which predict the shape of the fold with 90% accuracy. It's AlphaFold, kind of recent, big kind of achievement in this, and this artificial intelligence or deep learning mechanism. Kind of not very, kind of idea is very simple and very old. Was implemented with tremendous computational power, and now it was Nobel Prize. I forgot either in biology or in physics for that. Yeah, for that. People receive 2 Nobel Prizes now for artificial intelligence, for the deep learning, and one was in biology and one was in physics. This was in biology, right? It is a famous group of people by Baker who was working a lot for prediction of protein shapes, and the best they were doing before actually was human intuition. You look at the sequence and you just, by analogy, guess what will be the shape. Plus some computation will be a little bit, but now this kind of intuition replaced by matching pattern by artificial neural networks. But this is again, and this is not only for protein. It's called this way. The same, say, a virus is being assembled. Yeah, we have a virus and being produced as pieces in a cell, and then whoop, it's just being assembled and make very complicated particle. Or the ribosome, it's a complex, is containing a couple of dozen molecules, big molecules, and again, once you synthesize them, they know to find their places essentially because it's minimized some energy. But that it happens so smoothly, so you have a rather complicated, you see, it's highly dimensional space, and there is some gradient, and you follow the gradient, of course, stochastically, right? It's never kind of exactly gradient. Everything can kind of random motion. And then there's some, but this random motion, must say again, I never kind of managed to learn exactly what is kind of motion of molecules in the water. Yeah, it's not a gas. Yeah, what are the kind of freedom of a molecule in the water? So the distance between molecules with the same order as water molecule has about 3 nanometers in size. Yeah, it's 2 nanometers, and what is the gap between them, how they move? Of course, there is diffusion, but I don't have a good picture of that. So in the air, it's kind of more clear, right? Because distance between atoms is about 10 times the size of the atoms, right? Because air is 1,000 times lighter than water. So It exactly it give you 10 times increase in the linear size, and then it's unclear. It's clear how long it runs. Yeah, it's just it goes with the speed of a molecule. It's about 300 meters per second. How long it takes to heat each other? Yeah, you can figure it out, and you have microsecond scale for heating. In water, I don't quite understand it in a second, but this is again it's microsecond scale. The fusion rate and moving microsecond, but again, I'm not certain exactly what is 3 microseconds and 10 microseconds. And this is in a second you see why it's relevant. But this is again one picture one has to have in mind, but I don't have it good for water. I couldn't find it in description, simple description. When you take books on structure of water, it immediately write formulas. We take 1 page. Kind of start formula after formula, and also terminology I don't understand. But this is a water, of course, is very special. For liquids, water is, and on molecular level, is not unlike of anything else. And certainly, for example, what is energy of kind of this? So it all the time is divided into different complexes, not H2O. There is no H2O in the water. It's maybe HO, H or H3O, something, all this thing floating there, common and engage and disengage energy of an interaction about 2-3 kT. And there are computation experimentally, theoretically, but it's never clear when I read the article what it means. Yeah, I mean it's average, or I mean individual thing, or statistical, or whatever. So it's not very clear. But anyway, this is the order of 2-3 kT, and there is a rate of interaction, collision between molecules of order of microseconds, but I'm not certain how many. So one was weak interaction. Then there are covalent chemical interaction which change molecules. For example, we breathe, our oxygen goes in our lungs, oxidized glucose there, and there's a chemical process which goes in a very particular way. That is not burning, we don't explode, so everything's controlled by some enzyme, so it's enzyme-driven chemistry. Enzyme meaning catalytic, so it's a process which is being controlled and accelerated by catalyzed by enzyme particular proteins which were synthesized. In a second, we discuss what are the synthesis, and they kind of control the rate of various chemical reaction. And I was saying one of the fastest reaction is catalyzer, which when you throw it into hydrogen peroxide, so it's H2O2, it's kind of immediately see oxygen, molecular oxygen going from there, and it accelerates the rate by many order of magnitudes, but the rate there actually also in microseconds. It's maybe 1/10 of a microseconds rate of this chemical reaction. It's one of the fastest enzymes, and sometimes you see it's like an increased rate of reaction 10 to the power 20, but this because you compare it, so still it will be slow, it will be no faster than this one, because you may have reaction which normally happens in the, say, a million of years, and then you do it in a second, and certainly it's a big improvement, but it's not that fast, yeah. Still, and there are very slow enzyme, one of the main enzyme working in the world, which synthesizes, using photosynthesis, produces glucose out of thin air. It's Rubisco, and it's most kind of distributed enzyme on Earth, the most distributed protein on Earth, and I think it's about only 10 molecules per second. Yeah, for some reason it's very slow, because it's all the time poisoned by oxygen. It's kind of one of the major processes happening in the world. Yeah, and still one doesn't fully understand how it works. But if it stops working, we shall die, or we shall starve pretty fast. Yeah, maybe it depends on many things we don't understand. So how it happens, and so what actually happens, and what makes it faster? And so this picture repeats from book. So let me use picture. Yeah. So we have some process, and so we somewhere here, and then my picture is not good. Somewhere here. And we have to go from here to here. Here is our process. So all processes, of course, go downhill. right? We always consume energy, all they release energy. I mean, everything exothermic, and maybe endothermic, of course, if you add fuel. Yeah, but globally it always goes downhill, and then there is an obstacle. You have to go all these obstacles. Activation barrier. So I must say I don't understand why the barrier exists at all. So maybe you can explain it. You have a molecule, right? And so it's some quantum state. It's certainly quantum. It's some quantum system, and then it's not in the minimal system. So what keeps it? So it is not elementary quantum mechanics, electrodynamics, yeah, because when you go down, something burns, energy goes, so photons go out. So it's a one first step kind of in this Feynman diagram, which is first approximation Feynman diagram. I know these words, I don't understand them, but can you see in simple words what keeps it keeps it here, not in the minimal state, right? Quantum system tends to be in the minimal state, but it's not. It may spontaneously go there, and it's kind of clear how to measure this experimentally if it's really there. You can make experiments which can interpret in the height of this activation barrier. But how in practice, mathematically, physically, why it is there? Why simple quantum systems, a molecule like this H2O2, what keep it in this state, which is not minimal? It's not minimal energy, right? Minimal energy when you have H2O and O, well, eventually it will be 2O, yeah. O2. This will be this will be minimal energy, and this is not. What keep it there? How it can be there, right? This I must understand basic physics, and I failed to find any explanation in the literature. It's some
intermediate configuration too.
No, but we have some configuration again. But anyway, it's quantum system. It's minimal of there is some energy, but energy involves the word they say. Of course, not energy of the electromagnetic field. It also quantum electromagnetic field. Yeah, there are particles and there are photons, and there is a photon field, electromagnetic field. And this photon field is not stationary, and because it's not stationary, because it's defined by Maxwell equation. The Maxwell equation is involves time in an essential way, not like Schrödinger equation. It's always time dependent. Therefore, it is not kind of abiding Schrödinger equation. It's perturbation of Schrödinger equation, and you write some perturbation formulas. But I don't understand kind of before you write formulas what they mean. There is a quantum system. You have proton, electrons. It's not in the minimum, because there is a photon involved. And so what is Hamiltonian there, which is minimized or not minimized? So it has to have also have this, but you don't look at the whole quantum field. Of course, use some approximation anyway. I'm happy to see this approximation, but what is the Hamiltonian? It's never written. People write some...
No, Dirac or the Hamiltonian coupled to the photon field. So
yes.
You can write an Hamiltonian.
But why is this? What keeps it? What in quantum mechanics creates the barrier?
I mean, what keeps it is that the time to quantum mechanically you can always go inside the barrier, but this is exponentially small. Okay.
Yes.
If this is much larger than the...
No, but why barrier appears at all? Where barrier is quantum mechanics? It's linear operator. There is linear operator. Where in this linear operator there is barrier. There are eigenvalues.
No, but it just means that the wave function, when you go in a forbidden region, you are exponentially decaying. Instead of having a wave, for instance, the electron, when it is on the permitted region, its wave function is oscillatory, like a cosine. If one electron has to go through a barrier, the wave function becomes...
No, why there is barrier? Why there is barrier? You see, we have a linear operator. Abstract. In Hilbert space, there is linear operator. Where is barrier?
But barrier is a form of potential.
In the potential, if you take just ordinary Schrödinger equation, you can put a barrier.
No, but potential electromagnetic field, yeah? There is no barrier. There is no barrier.
There is the Coulomb interaction of all those...
No, there is no barrier there.
No, but there is atoms, and so they create some configuration of the field, and there can be some barrier, which is specific to these molecules.
So you say there is a barrier in the electromagnetic field before any photon comes?
Yes.
Because in order to go, you have to involve electromagnetic field, of course, quantize electromagnetic field.
Yeah, but the Coulomb field you don't need to quantize.
No.
The Coulomb forces between charges, okay?
No, but what is again? I understand there is this field, there are charges, but from quantum mechanics, there are some axioms, right, that you always say in sitting on some eigenfunction on the minimal eigenfunction. What is the physical interpretation of this mathematical stuff? I can imagine there is mathematics, but what is the physical interpretation? How you go from quantum description of operators, how you go to the picture on the real world? This is what I don't understand.
But it's not necessary in the minimal quantum state. It depends on in the minimal eigen. It can be in the other state. Yes, it depends on the.
No, but you know, we in the world of quantum mechanics, we forgot where it came from. What you have is operator, and there are spectrum, and right. So you say, aha, you're sitting on this eigenvalue and this low eigenvalue. So this where barrier come from? Where the barrier come from?
No, but in Schrödinger's equation, okay, you have the.
No, forget about it. I have operator. I have already quantum field. There are operator.
Is the kinetic term p squared over 2m plus a potential v of q. If this potential has this shape, okay, then you have approximate stable states which are inside the barrier. Okay, if you have like in a ball, okay, and you put a particle in the middle. Schrödinger equation is a good approximation, and these states will be metastable. They will tunnel through the walls. Okay, so the potential here is created by the Coulomb interaction.
So there's this Coulomb interaction create this barrier.
I think so. Yes, because you don't need on-shell photons for this.
But how you know? I have this molecule. Look, I have this molecule. And I know how it is, and from that I can compute this barrier. Is the shape of this molecule or what?
Yeah, I think it's shape of the molecule. For different molecules, you will have different shape of the Coulomb field, and some of them might have this shape.
For instance,
okay, so it's the shape of Coulomb field which creates this barrier for the particular molecule.
Yes, you take, for instance, a certain number of charges, and
okay, what does it mean shape of a molecule? I'm sorry.
The distances here.
Yeah.
Just distances between.
Compute the Coulomb.
Coulomb of nuclei. Coulomb generated by nuclei. So you fix the nuclei and take Coulomb with respect to them.
And fixed position in space, you compute the 1/r potentials between all the charges. And then you explore all possible configurations where they are close or one of them is very far. It gives a landscape of a potential as function of the geometrical shape of this. And
so it's a classical picture, just classical picture of potential.
Yeah, in the Born-Oppenheimer approximation, you can start doing this. Compute.
Yeah, I know. In this approximation. Yeah.
And then you say, now I quantize the system, and I say my things become.
Okay, no, don't, even before quantization, you say this shape is actually a classical shape of the potential. The question how you go about it. Okay, now, okay, good. I feel. No. This okay. That's so it depends on the position of between nuclei. Okay. So okay. Then you can forget about quantum mechanics and everything is fine.
Misha, but are you sure that you can have this molecule be stable? I think if it is in a pure condition, it's are not stable at all, and it's stabilized in this shape when it is diluted by water. So because of the communication with water.
No. They are stable without any water. No. If you take PY, we have this H2O2, maybe just abstractly.
If you take it in normal room temperature, it immediately by itself.
No. This molecule, no. Most molecules exist metastable. We all made a metastable molecule.
It is stable only under very.
No. But barrier keep it stable. It's about 50. For this particular molecule, but this barrier about 50 kilocalories per mole. It's very stable.
This molecule, it is stable in.
No. Water has nothing to do with this at this stage. No. On the contrary. So how it may be destabilized? Water may destabilize it because some molecule in the water would fast enough, it have enough energy to kill it and contribute the energy, and then it goes to the stable. No. Presence, of course, depending on temperature, of course. This is how you actually, because the point is, another point is, how you measure that. When you look practically measurement, you using this kind of chemistry, you see at what temperature it starts reacting. You raise the temperature, you see how it reacts. And then you interpret as a potential barrier. But that's, in this picture, deceptive, maybe not a barrier of energy, it may be something else. And this we shall discuss here. This, from the moment we go into classical, everything clear. I didn't quite realize that this barrier refers to the classical potential field, right? Because it's actually this quantum process. Of course, this tunneling process, and we don't know how relevant it is. It's a big discussion. In enzymology, tunneling is essential. Now people, lots of articles saying it's essential, but some disagree with that. But now let's look at this closer. So, but how it happened? So you have the barrier anyway, and so you need certain energy to go. So you need some fast particle moving in water, and they come very rarely. You have to wait for a long time before it comes. You can compute it if you understand water reasonably well. Actually, also I don't quite know how to understand it. What's the fluctuation of the velocity in the air? It's kind of clear to me. I see how this Maxwell distribution works. In water, it's not clear to me. What is the fluctuation? How long you have to wait to have sufficiently fast molecule of the water, to having energy enough to pass certain particular barrier. Because you see, I never saw this, because this is very easy to compute. To find this shape of this barrier with electromagnetic field, it's elementary kind of stuff. It's not Schrödinger equation. And again, I never saw it actually being in any papers I looked at. But granted that at least conceptually become something clear. But then interesting point, how it can be. So you have some other molecules, right? And then you pass a barrier. So how it can be, right? You add and see this protein molecule is shaped. Internal energy of this is negligible. It's a few kT. Proteins kind of rather soft stuff, yeah. So you take in your hand, you have to take a stone. You can't break a stone having in your hand. It's really energy, but by order of magnitude more, right? Especially we speak about in terms of affinities and time scale, yeah? So how it happens. And then which I read, and I couldn't understand it, but now I think I can interpret this. So this is idea going back to Holden and then to Pauling, saying, aha, there is some intermediate state. When you go from some molecule, you go to some other, but there is some intermediate state. It may be virtual. It's not actually something, but it's some intermediate state. And certainly it's not one-step process. It's many-step process. And your protein has such a shape that it binds. It has a shape of this protein such it sticks to these particular molecules. Now, there are two cases. Before you have to look at another case which is more transparent. For example, how you can force burning of, and the room temperature of burning hydrogen in oxygen. And actually there is kind of interesting, fantastic story, detective fantastic story by Isaac Asimov about that. If you bring in the mixture, you have kind of, say, balloon with hydrogen, and you open it, nothing happened. But if you bring the platinum, especially if sufficiently spread, yeah, then it burns. Platinum catalyzes reaction, actually for other processes. Not only platinum, many metals do the same, yeah. By the way, the basic chemical process what you use on Earth and in industry is binding nitrogen from the air for fertilizers, and it is done, which I shall say more about the lectures. We're using some catalyzers now. You use iron for some reason, but anyway, metal absorb molecules of different gases, for example, oxygen and hydrogen, and bring them close together. And this is a weak interaction. They stuck together, so it means kind of geometrically some potential bringing them in. So there is kind of a region when they come together and they move in this region because under kind of gradient of this weak attraction. And because density tremendously goes up, probability of hitting one another with sufficient speed to overcome the barrier becomes by far smaller, and so it starts burning, right? So this way you can produce burning of them without matches, burn hydrogen. I'm not certain happens in the air. Maybe even in the air, not necessarily in pure oxygen. Hydrogen will burn. But that's clear. So you don't change energy of the picture. You kind of change entropy, right? You bring them together. It doesn't require any energy. Like you have a key, you can modify. You have a wrong shape key, now you adjust it. You have right, wrong key, and you open it. Or you just, if you go on the information level, not on the level of physics, you can actually have, you have number key, and you have, if you know the right numbers, you can push them, yeah? And this, of course, not physically inexplicable. Why you have particular number on the lock, you put them, and then it opens. So you create physical process out of knowledge, yeah? It's something incredible, right? It shouldn't be so. It's a magic, right? How you can explain physically? You have some physical process, open the door. What you do it with your knowledge of some number. You put this number has nothing to do a priori with any physics, and then you open the door. It's impossible. Right? It is impossible. It's magic. It happens, and this life. This what about why life is not explicable in physical terms, yeah? Because it's magic. It's information. Right? And it's quite interesting as to unravel this mathematically. So what happened mathematically? Yeah? This we shall come to that. But now let's come to enzyme, because I think I was very kind of happy when I understood something. But how? So you have this thing, yeah? Now what happens when you add some protein or whatever? You add new parameters to the picture. So you have, of course, first of all, even this picture is cheating, yeah? It's highly dimensional space, and there are many parameters, and then there is a kind of mountain range, a potential. And this mountain range is with complicated shape, and what is their height and what is the barrier is kind of very unclear. So what if you have multi-dimensional range? What is the height of the mountain? Right? You cannot measure it. You only make statistically. You see probability of going over this barrier depending on the temperature. I think how it's done experimentally, it is a Van Hoff, I think, formula or somebody's Arrhenius formula. You see how process probability of starting with a certain temperature. But anyway, pretending that. But now you add new coordinates. First you add coordinates, poop, so you have just everything spread in many dimensions. And then you slightly perturb it because you added new energy. But your perturbation is very small, right? There is no energy in this protein, right? It's not a string inside, nothing inside. So you slightly change it. And why this slight change can tremendously make a reaction much faster? Because you changed a little bit, yeah? So what you can do, for example, right, in this kind of picture. So you had this picture, yeah? And then goes down like that. And you change it a little bit, so you make it like that, but I understand what you do it like that. This was the original one. And you have a little change, you make it kind of in this way. And then the reaction may go much faster. And the answer is, it certainly goes faster, but still goes incredibly slowly. It still goes at most in this, as enzyme work on the scale of microseconds, maybe milliseconds. When you say it's 10 minus 20, it just, which was confusing me. So it makes this speed of reaction by so much. It's because if you measure it with years or millions of years, it becomes very fast. But on the scale of seconds, it's still slow. It still happens on the scale of collision between molecules. But chemical reaction, which coming in this kind of quantum world, they go on this speed up to femtoseconds, maybe a million times faster, or at least 1,000 times faster, right? Or at least it's picosecond. So this intermediate state, if they exist maybe picosecond time, and then disappear. But now, so what happens? With my understanding, again, I never read it in textbook, but certainly because everybody probably knows that, and but that reason is never said, right? I remember when I came to France and I was asking some questions about phonetics and said it never written in textbooks. I said, okay, well, everybody knows that, yeah. And people will say, why are you asking? Of course you don't write that, yeah. In the same day, physicists apparently know that, and they never say it, and biologists, I guess, don't know that, and so they don't write for that reason. So it's empty space, yeah. So what happens, that you need certain energy to go from one state to another. And then probability, but you have only kind of relatively slow molecules, and they cannot help you much. Then you say, well, having two slow molecules, it may be the same probability as having one fast, but only if they come simultaneously. But there is a big difference in time scale. Molecules come on the rate of microsecond, and this thing a priori exists on the kind of picosecond. But here, if you have that, when you stuck to them, they keep a little bit longer. This intermediate state, instead of survive for 1 picosecond, now we live for 1 microsecond. And then there's a chance second molecules come there, and another come. And then and this still on the same scale. 10 of them, you don't take 10th power of that. If they come synchronously, the time goes by factor, multiply by factor of 10, 10, 10, 10. But here only multiply by 10. So it's only make it slightly slower than microsecond, the collision rate. So it's still, and so you achieve this maximal speed exactly of the collision rate. And so this is maximal speed I understand for this, the best enzymes. But I'm not certain I understand correctly because I never saw it written. But this is one thing, obvious explanation. Right? So it's very essential that there are very different time scale in chemical reaction, which you go up to femtosecond, usually I think picosecond at the right time scale. Anyway, nanoseconds, everything happens. So it's 1 billionth of a second. And then there are microseconds of the diffusion rate or all these rates of molecules in the cell. And then everything comes to the same scale. This is my understanding, this intermediate state and all this talk about intermediate state. So it's one explanation. And this actually would be interesting to have purely geometric description of that, which I described. Of course, nobody showed this picture on this. They just say this: you have the intermediate state, and it is known in certain instances that the loss of intermediate states, even for burning of hydrogen and oxygen, there are about 1,000 intermediates, and there are some experimental data for that. And they all extremely, of course, live their life is on order of nanoseconds or picoseconds. And you read all these words in an article I say, but none of them give you the full picture because much of this is still unknown. This is all hypothetical. And also, for example, this energy barrier. It may happen this barrier is zero, no barrier. It may be just purely entropic because you may have a wall and there are some narrow kind of bypass of this wall, but it's so narrow, so entropically they're very unlikely. And that's another possibility for what an enzyme can do. It may just change the channel like it happened for synthesis, when you bring two molecules together. And the same it can do in there. It just can position, and if you have this puzzle, you have to disengage two pieces, it's very difficult, you cannot do it unless you know it. And protein know that and bring position, so when then random motion has much higher chance to disengage them. Again, if this happens or not, you have to understand much better geometry of these molecules and just imagine what happens. And I believe it's unknown in this geometry. It has not been done by anybody in principle, so we can verify it. But this is another possibility which may happen. And again, it's said in books. It may change, may find different paths. So whatever it means. Is it possible or not? It's unclear to me. So it depends on the possible shape. If I understand what correctly you say, it's just the shape of the electromagnetic potential. And this shape you can kind of analyze. Right? It's kind of elementary mathematics. It's made difficult computationally, but mathematically it's rather easy.
No, but I should correct that. For as soon as you have covalent bonds.
Yes.
So it means you start with the geometric positions.
Yes.
But then you need to solve Schrödinger's equation for the electrons that give the covalent bond. So they give a binding energy, which contributes to the potential for the geometric configuration. Okay? So the potential as a part which is classical Coulomb plus quantum solving of the... Okay. And this is then the landscape of the potential for the molecule. Okay.
No, but Schrödinger just add kind of kinetic, integrated kinetic energy in Schrödinger, Laplace operator. Not Laplace operator, no, but we have...
The orbitals of the electrons.
No, but what could you add Laplace operator to that and see what this kind of how the spectrum behaves?
No, but you have you have light particles and heavy particles, okay? The
Yes.
heavy particles are what you see here, HO, okay?
Yes.
Then when you
I see. Oh, yeah, you have to find the right position. Yeah. Yes. Of course. No, but it depends on the distance.
are light, yes, and the solution for
Right.
this gives a potential now for the positions of the heavy
Yes,
things.
yes.
And this gives the landscape, and this gives this barrier.
Approximately, yeah. I agree, but still but have to adjust the distances. But anyway, you can imagine that there may be this kind of narrow passages. Especially when you add more, because you add this extra molecules, and they have quite significant electric field there. Of course, this electric field distorts this picture and may create this, or may not. This is how complicated this landscape can be, because the principal component is still now just sum. It's a sum of Coulomb potentials. And then there is third kind of explanation. Actually, in book they say force. Yes, no. I said it will be one is this distortion of this field. Usually, I mean, what you see, the point is they repeat the same thing. In most things, it says copy and paste, copy and paste, copy and paste. Yeah, and so they repeat the same thing. Obviously, the authors don't know what they talk about because they repeat it. You know, there is principle by Haldane. If something is repeated, everybody knows that. Yeah, I forgot how he said it. It must be nonsense. Yeah, if you refer, everybody knows that. It must be nonsense by definition. He said it in a more kind of kind of style English language, more idiomatically. But and from my understanding, most people who write this usually on the internet certainly keep repeating it. Yeah. But because geometry, it's indeed something maybe happened, highly non-trivial geometry there, which I don't think has been analyzed. And the third point is that it may happen that what contributes to this is vibration in protein. When you have a molecule like protein, it certainly oscillates all the time. And it oscillates on a pretty fast scale, maybe even slightly faster than it may be a rate of oscillation maybe on the picoscale, not on the microsecond scale. Right? Because it's protein, there are covalent bond between molecules, oscillate, and then there are weak interaction, but still energy slightly above the KT level, and so protein oscillate. And then it's sitting in the bath of molecules, and so it certainly total energy is just the number of energies there. But oscillate much faster than on the microscale. Therefore, fluctuation, that the fluctuation of some very fast moving atom there is higher than would be in the ambient water, because the time scale is different. It leaves 10 times or 1,000 times faster. So its oscillation is 1,000 times faster than collision rate for molecule in the water. And therefore fluctuation appearing in a fast motion will be also 1,000 times faster. Therefore probability of having some molecule moving very fast there higher than in the water. Is correct for example, in metal. Yeah, oscillation of atoms, right, is much faster than collision rate in the air, and therefore probability of fluctuation accordingly much faster. So having pathologically fast in this Gaussian distribution, probability going here per given unit of time making much higher. Of course, it moves much smaller amount, yeah, because speed are roughly the same, but displacement accordingly smaller, right? And the question is, even this small displacement is sufficient to hit the right place in your molecule and produce this passing over the barrier. And again, this people say in all these words, but I don't think any kind of convincing argument that it is true. How fast how close you must come to particular electron, to electric field, to whatever, to create, actually this is a quantum jumping over the quantum barrier in the quantum mechanical step, and photon goes out, right? So it's really quantum mechanics. It's not some. So you have to understand better than I do, and I couldn't find it. But anyway, that's a purely kind of classically, you can kind of imagine this model being developed to pure interesting mathematics. So there are three different phenomena, purely geometric, which probably happens here. You have to interpret them quantum mechanically, but none of them was mathematically studied with any degree of precision to see if it fits a real measurement which you can do. But even theoretically, it's not so clear. So one is that you create the Ras intermediates. So you just change this scale, you produce little wave, right? So you can, several heat will do. Another one that you have vibration in protein. And thirdly that you change this. So what I said, yeah. I said 3, vibration, proteins, changing this in intermediate state, yeah, and creating this electric field in the, also from the protein in there. So there is all the things, and they all said kind of, in words saying this may happen, and the only thing which definitely has been studied in the intermediate state exists, not always, they say. Sometimes there are intermediate states, sometimes don't, and then it's unclear what happens. But my understanding, still people admit they don't understand what happened there fully, how enzyme works, and that's essential. If when you start understanding that, it may be essential for industry, because very many industries depend on using enzymes, right? Like yogurt, which you have, or beer, or wine, everything is done by certain enzyme, and now it's enzymes done artificially synthesized, using usually bacteria or something, not directly, of course. Yeah. So this is a story about enzyme, and very, very superficial one, and huge literature about that. I tried to read some and got a little bit lost. But this is the one things which happen in cells, and this is enzymatic chemistry, and this enzymatic chemistry is involved in particular in production of proteins. So I said there are one thing, it's self-assembly. So you have some molecules, usually large, and certain particularly charge distribution, dipole distribution there, and then according to that they fold and make some shape. And this, of course, will not happen for any distribution, but for very particular ones, and evolutionary nature found this good configuration. And nobody, of course, understands how typical this is. They're very rare. This configuration, of course, are very rare. For that reason, I think physics can tell you nothing about that. You can say what happened for genetic one, what happened in principle, but these are like you have instruction how to build a house, and physical analysis of what you wrote, kind of how to build a house, and not about physics. Of course, you follow the role of physics when you do that, but this particular sequence of events are very, very special, and certainly relation between the text when it's written and the implementation is very tricky. And this is one of the problems we have to face.
And are you saying that the protein can do its functions only when it is folded in a unique shape, or
in a particular shape? Each shape specific for the function. Yeah, it's very this enzyme are very specific. They only accelerate particular reaction. Sometimes they may accelerate several similar reactions, and this apparently used sometimes in cells. But they're extremely specific. Yeah, and certainly, and therefore the way they are done is like you key to the work. You are adapted to a particular situation, right? And again, you know a posteriori how it works, but evaluating and deciding what kind of function this enzyme will have by looking at this formula, even at the shape, not so easy. And the best, apparently, and people try with, again, with new artificial neural network, by just looking at many examples, looking for patterns without understanding what happened there. And this is not accidental. Why so?
But are you saying, sorry, I don't know, enzymes are crucial for the protein folding, or it's a separate problem? I thought this was a...
No. When protein folds, enzyme crucial, but on a stage of production of protein which I didn't say yet, right? All chemical processes in the so you create first shape by using enzyme in particular proteins, which have particular kind of shape, particular position of different amino acids, and so a certain distribution of kind of dipole, and then they stuck together according to that, and then by themselves, just following that. But the fact it happens in this canonical way only possible because of very special sequences are used. Most sequences will not fold, and actually even real world proteins, half of them don't fully fold. It's kind of only half of the protein really make these nice definite shapes. Some globular proteins, and still often there are some free parts which don't fold, and this is rather unclear what happens with them, how they function. At least my impression is being somewhat controversial also. So what happens, what is their function? Again, people say some genetic words, but that you can say anything, yeah, without really true knowledge. But now proteins themselves are being synthesized, and the synthesis of protein involves two top processes. First, it is going from DNA to RNA, and this is a kind of simple on one hand process. On the other hand, it's not so simple. It's logically simple and kind of still chemically sophisticated, and it's not truly chemistry, because besides chemical reactions, so we had two kind of processes, and the fundamental process in the cell, so self-assembly. So we have some kind of molecules, or several of them, and if they have some particular charge there, which kind of called usually hydrogen bonds, they stick together and take very particular shapes, and then they can function in this shape. And there are some exceptional case when this is done spontaneously. It doesn't use particular configuration of the charges, but uses only kind of symmetry, and this is how membrane being assembled. So I was giving picture. So there is a membrane of the cell. It also self-assembled, but for completely different route. So it's made out of these molecules. So these molecules must be there, or synthetic. They're small molecules. They're lipid molecules, and this is usually bilipid. So there are one from here, another from here. But this kind of a material, and this molecule, and they stuck together and make a membrane. And they do it by also kind of self-assembly, purely physical process. You dilute, we have like soap films, and if this molecule are distributed in the water under certain temperature and certain density, they assemble and make this kind of nice surfaces. And this is, the reason is that not because they stick to each other, they're actually completely slippery, and this little thing make kind of a two-dimensional liquid without friction. They move completely freely in this direction. There is no attraction between them. But in this way, they minimize free energy of combination, this kind of molecule and the surrounding water, right? Because they don't, they're bilipid, and so some of them have interaction with water molecule and some not. And in this way, because if they would be free at certain temperature, they would interfere more with water, because water want to stick to each other, and if they separate it, they have more obstruction for the water to the different OH kind of ions to get stuck, and therefore in this way they minimize the free energy. People say it's entropic, yeah, of course, because when you write the whole model, temperature, entropic term is involved, but certainly it's energetic, yeah. It's free minimization, free energy. Like when people say it's entropic, it's still free energy, of course. You minimize free energy.
But free energy is energy minus
yeah, of course.
Entropy is...
So entropy is enters, but of course, because when you look at this term, yeah, there is temperature which enters there, so there is entropy. But anyway, this is quite remarkable process, and it's quite really mathematically very remarkable, because these surfaces they use to satisfy some nice differential equations, right? So this membrane is believed they have minimal bending energy per given area and volume they bound, and this is seen for erythrocytes, which have this nice bivocal shape, yeah. Rotation is symmetric. This is our erythrocyte, how they look like. They are, of course, very special cells. They have no nucleus, no DNA in them, they're not true cells. But when I say about every cell has that nucleus, something, DNA, it has no DNA inside, yeah. It's only proteins, and it's hemoglobin molecules, yeah, and this has this shape, and this is mathematically unproven. So if you look surface in this space or given area and given volume and bounds, then minimal bending energy, so it's integral of the squared curvature, minimized for this shape. And so, my understanding, people who worked on that say they proved it, but they proved it, of course, assuming the symmetry, rotational symmetry. Like engineers, they wouldn't care to prove about the rotational symmetry, but they believe it's unproven. So I never saw it proven. It looks to be highly non-trivial, kind of, well, for them maybe a dimension thing, not so difficult, but this makes sense in high dimensions. And again, this also makes very interesting mathematics, because if you usually, when you look in the minimal surfaces, they look like static entities, and corresponding differential calculus, your perturbation, yeah, when you look analysis, you just look at this kind of continuous perturbation. But in truly, it's a stochastic thing, yeah, and they can break it, and so they actually change, yeah, on certain, or probably on the I think on the nanoscale, nanosecond scale. So the topology is not fixed, they can really move, and still they can breathe, but still they minimize something. And this would be very interesting to have such a mathematical model, including them with temperature involved. So when temperature goes up, they dissolve and then they can come back together. So it's by far more sophisticated mathematics than usual theory of minimal surfaces. It's really much more structure there, and which may be essential for understanding partial kind of elliptic equation of this type. They are not. They have this stochastic interpretation, and everything is stochastic. Maybe also some quantum also. But this is a remarkably interesting structure, and this is about 3 nanometers. Yeah, the cell, as I was saying, the thickness of the membrane about 3 nanometers, and relative to the size of the cell, it's like airplane and its boundary of the airplane at the same kind of level of scale. Okay, let me make now little break. What's said by this, by Diderot-D'Alembert. This actually, this period, Diderot-D'Alembert, so Lavoisier, what at this time, and Condorcet, and Buffon, they were extremely smart guys. They were a company of extremely kind of intelligent people. I don't think ever, I don't know where it was happening before such a constellation of smart guys, yeah. And so what about an egg? So again, from physical point of view, let's look at an egg, and we have a rather homogeneous, kind of amorphous mass. It's not fully homogeneous, but essentially homogeneous. And then you just keep it at a right temperature. I think about 30. I forgot what is the right temperature. About 20, 25 or something. And then kind of chick comes out of there in 3 weeks. And how could it be? So if you make any kind of sample of this egg, forgetting minor kind of asymmetry, it's fully homogeneous and rather amorphous. And then we have, I think I mentioned something already last time. I want to elaborate on this more. That how could it be? So why it looks impossible and why it is possible, despite the fact it kind of looks, it seems impossible. Of course, the first thing, when you sample it, yeah, you miss the essential. So first, imagine your sampling is representative, as you do everywhere. There are, of course, billions of cells, and you make million samples, and they all look more or less the same. And so you make your conclusion that all are the same, right? If you have billion objects, you try million, they're all equal. So they assume they're all equal, right? That's major principle of science, right? If you measure millions of times out of billion, they're all equal. They all must be equal. Why so, actually? Equality is a very rare phenomenon, right? So if you have a rare phenomenon which you don't expect to happen, and it happens, you say it always happened. this basic principle of human knowledge, right? But here it doesn't work. So biology is illogical. Basic logic of science unapplicable here, because there is one particular cell, right, which is special, and this one cell from where everything comes, right? And this is so imagine in physics you do that, you look atom here, all the same. No, but there are one special, and this special determines what happens. It's certainly very strange, and but this is so. So first, if you kind of believe in physics, a kind of usual kind of logic of science, this never happens. It cannot transform in a reasonable amount of time, kind of reasonable saying it may take millions of years, but not 10 to the billion years. So it cannot turn into anything structural. And kind of there are, of course, energetically nothing happening, so everything right, because there is heat being produced when egg develops, so entropy goes up, everything is fine. But still there are two things which kind of happens, they are closely related. It's a break of symmetry, stochastic symmetry, and increase in complexity. And so naively, quite reasonably expect, none of this may happen. If you have something symmetric, and you go on, and there are symmetric kind of life, basic laws of physics are symmetric, symmetry cannot be broken. And this partly related to the fact, of course, that entropy must increase, because it's stochastic symmetry, and you believe kind of there is some averaging in time when there is enough randomness, which increases, of course, entropy. And for the same reason, you cannot have structure, but you don't know what exactly the structure means, but complexity definitely goes up. So again, in what sense, again, complexity is, again, not completely obvious how to properly define it. And so what is this complexity then? So again, this is an essential part. Everybody says structures of life are more complicated and more complex and simultaneously more regular than things non-alive. And again, when you say these words, you have to know what you mean, right? And complexity here, mathematically, it is a kind of a generalization or kind of variation of Kolmogorov complexity, though you must be careful exactly how you define it. Because Kolmogorov complexity and expression is purely discrete thing, applied to discrete structures, say discrete sequences of 0 and 1s, this is kind of a typical situation. And complexity of such strings is the maximum length of a program in a particular kind of style computer, some particular Turing machine, is the length of the program needed to represent this sequence. Minimal. Yeah, it's a minimal. It's a minimal, right? It's a minimal, absolutely. It's a minimal, right? It's a minimal possible thing. And this is a kind of very non-efficient definition. It's incomputable. One can prove this in principle logically incomputable entity, because this program may be extremely long. We don't know how long. The time functioning of this program may be very long. Program may be short, but time going may be very long, and it may be really uncomputably wrong, right? Maybe not non-computable function as a function of the input, and therefore it's kind of not very well. In real life, you never come to this situation. It's one point, and from this point of view, the most complicated sequences are random sequences. You take really a random sequence. This cannot be. There is no way to shorten it. Yeah, there is no way to make it shorter. And only shortening appears when there are some kind of secret periodicity. Some part of this may be similar to another. You may encode this similarity and then make it shorter. This is what you do, say, for languages. By the way, nobody knows. I mean, people speak about that. What is the actual complexity of the human language? How much it can be condensed? And it's kind of, it's not, actually Shannon was making this experiment, kind of entropy of the, and then Kolmogorov was repeating it, what is the entropy of the human language, how much you can throw away, and then still guess what is there. And now much of the artificial intelligence analysis of text, following in these steps, yeah, seeing how much you can fill in some gaps in the language.
You mean the complexity of a given text or the language as an abstract?
Well, depending of given text, yeah, of given text, of given string. And so what's interesting, I remember I was actually present on a lecture actually by Kolmogorov. He was giving a lecture about that. And so, at least in Russian, if you compare poetry, which is highly organized in Russian, or you take something from newspaper, which look to me completely algorithmic, Russian newspapers, as much as actually working, and then conversation with just people. And the highest entropy was for poetry. It's much harder to feel in poetry. And the lowest entropy was conversation. People saying all this is more or less all the same, all the time. How are you, etc. And this is, well, of course, very short interval. What happened on the long scale, so what we do know that there is probably deep similarity between texts of certain literature. Say, actors who learn kind of Shakespeare will be then very fast learning Molière, right? This is something very common. But if you they take technical text, the memory will have to start everything anew, right? And for us also we can easily remember pieces of mathematics because we are adapted to that. The same, you know, for chess parties, when especially clear. Chess masters immediately remember instantaneously party being played, right? But if they show random position of pieces, they can't remember that.
Right. By the way, a professional pianist told me that pianists never play four last pieces in concerts because they cannot remember it. Why it is very easy to remember Chopin, things like that.
Right. Yeah, there are these, exactly. There is this is kind of, what is a hidden structure there? And maybe related not more to our mind rather than to the structure or something else. Well, we don't know, but this is interesting, definitely very interesting issue appears here all the time. And so, coming to the egg, coming to the chicken, so one thing which has happened there, that there is this chicken zygote, and by the way, see, it is not completely one molecule, but has many, you know, much more chromosome than we have. For some reason, birds have more chromosome, but still, and this is not accidental, still a reasonably small number. All other basic molecules appears in much more copies. And so this is, again, typical feature of life structures, if you, especially if you take something very formal, like genomes of bacteria, and you have huge number of bacteria in some environment, they have all really identical sequences. They have sequence of millions of digits, binary sequences of length roughly million, and it appears in quadrillion copies. Well, in quadrillion copies, I think there will be some, you know, 1 in a billion will probably have mutation. Yeah, my understanding of the correction rate or like that. Yeah, is it correct? I think that you No, I think 1 in a billion if proper correction. But anyway, they will be essentially identical sequences, repeating many times, which is absolutely impossible from any physical point of view. But anyway, it happens, but the number of copies of the DNA is here, the number of connected copies not so many. And each of them appears maybe 4 times. Yeah, there are 2 pairs, and then from father and from mother, so it appears 4 times the same sequence, essentially for keeping it, minimize the error. But even the connectedness is rather high. We have about, I forgot, 17 or something chromosome, and chicken have so many. But on the other hand, the number of parts, kind of cell in the chicken is much bigger than that. It certainly goes in billions, and the mutual position, so the full information is huge. But it's encoded only on this DNA, and is comparable in size with ours, about 2 billion letters. So, and this what makes kind of ourselves possible. This tells you something about our complexity. It's the size of the genome give you upper bound on our complexity. But then, but in what sense? Well, certainly there are much more to descriptions of structure, but much of this structure is noise. It's just some random thing, and it's not being controlled by DNA. and some many random influences, and this because this big kind of discussion between nature versus nurture, what determines ourselves. So with all this random thing to say it's nurture, it's not in DNA. Of course, everything in DNA, and there is random. There is a random noise, and then you have to modify accordingly Kolmogorov complexity in saying, you have some. There are several ways to say it, but I never saw it really systematically exposed. There are some articles of this type, but not sufficiently kind of systematic. That on one hand you say you are not encode your sequence, but you like your body, your organization, but some random law which describes it. But when I say random law, it's tricky mathematical object, So it's some measure on some space or whatever it is. And it's not exactly when we can speak about Kolmogorov complexity, usual Kolmogorov complexity, but for this random law. Or you can say you have a Turing machine, but also which allows you random number generators. And how long will be programmed using these random number generators. But machine, of course, which implements all that, the Turing machine in biology, this I would say it is this self-assembly, it's controlled self-assembly. So you have a program written in DNA, following this program makes sequences of proteins, and boom, boom, you throw them and they self-assemble. And the self-assemble is your machine. But we don't know how it works. I mean, in principle, I think this kind of physics is kind of in principle known, but to make any computation, practically follow it impossible, right? It's absolutely beyond any computational possibility, and you have to bypass it somehow to find a way how to bypass it. But this again making this mathematics quite interesting, If you look at the logic of this, very sophisticated, interesting logic. There are logical steps, and I've done one which I kind of didn't say just before I wanted to emphasize. So there are chemical processes, there are self-assembly, and also there are motor proteins. So in particular, when you have synthesis of, say, even in the course of translation, so there is RNA polymerase, is a big kind of machine, of molecular machine, several enzymes involved. I had some here picture, but not in there, and it goes along and makes kind of synthesis. But going along is another process, and this happens in the cell all the time. When you have motion of one molecule with respect to another, and it's forced motion, so you go step by step, and you stick to this using weak interaction, but achieving step to have direction. Weak interaction involves no energy, so it's symmetric. You can go this way or this way, but no matter you want direction, you have to spend energy, right? To make something definite, you have to spend energy, right? Everything without energy is completely time symmetric and space symmetric, right? This is perfect kind of perfect balance, in this in physics, and then you have to use energy, and there is a way how energy is being used, and this is three basic things. There is a self-assembly, there is synthesis, molecular synthesis. Also there is also degrading molecules, but it's kind of not that crucial, essential, but not so crucial for synthesis of proteins. And then there is motor, proteins which moves there. So there are three things, and they all depend on one another. They're all interconnected, and everything depends on which order things made and what are the time scale. So it's an incredibly involved and confused thing, And so, but anyway, everything is written. There is kind of how it's done, and logic of this quite amusing, because as a result, chicken can produce another chicken, or one egg produces another egg. Depends how you look at this, by the way. Who produces whom? This is self-replication, and it was modeled mathematically by von Neumann. There is a von Neumann model of self-replication, and so this time was. So I don't want to say 2 words. And this is a kind of automaton. The first model of von Neumann was quite transparent, but harder to formalize. You have a kind of automaton, finite automaton, goes around, it takes some pieces, and then makes itself. And then he kind of formalized it using cellular automata. You have to formalize it, become tremendous mess. You have to everything describe in precise terms what happens, and there is a description and impossible to follow. Right? Everybody can grant this idea, can make his or her own formalization of that, but it's unreadable. I mean, in my view, it's kind of absurd activity. It's clear for mathematician you can formalize it. But if you start point by point formalize it, will be very lengthy and very unpleasant. However, life doesn't do that way. The way it's organized is not this way. So, of course, the major point of the description of phenomenon, it was before DNA was discovered, that part of this automaton is a program, just a string of symbols. And there are simple operations that it can make. Given any string where it describes some automaton in the language of this automaton, it can produce this automaton. Right? This simple thing, it's because it's universal. You don't have to be very special. This is like universal Turing machine. There is one kind of simple automaton reading something. It can implement in any program because what's involved this program and the program into which this program applies. Which again, I must say, there is something there mathematically not quite satisfactory because there is something completely non-canonical in describing a Turing machine. Right? It's kind of not fully mathematical, as actually physicist Deutsch justly remarks. This Turing machine is a describing physical kind of process producing computation. And then he is motivated by that. He said, now take quantum mechanics and do the same. Yeah, this quantum computers. This is one of the source of quantum computers. Indeed, this description in intuition is kind of mechanical. And mathematically, my understanding, you cannot in principle define what is computation. There is no satisfactory definition of what's computation or universal computation. There are several models for that. Recursive function, Turing machine, this matrix by Markov, we can produce billions of models, and they're all equivalent. Again, nobody knows what's the equivalent. Yeah? You say they're equivalent, but it's not mathematical equivalence. It's just something, right? Nothing is rigorous when it comes to logic, mathematical logic. Nothing is rigorous there. It's just lots of words pretending they're very rigorous, but usually they were questionable. This is kind of. I think it's paradoxical state. Logic is supposed to be most logical, but it's least logical. Yeah, it's of all sciences. But anyway, here it's very different. Yeah, the way there is no one kind of machinery. There is no one automaton. There are little machine. They're all very similar, proteins or RNA, and one DNA kind of where everything is written. Yeah, so a machine of phenomenon, once you have this thing, it can reproduce anybody and then reproduce this program. So it reproduces everything. So it's a universal computer. You cannot make exactly producing itself, but it may produce everything. And cells, not quite that, by the way. Yeah, they're not universal. They can do many things, but not everything. Because they can produce not only themselves, but can produce viruses. So you can look at them this way. They're a machine which can reproduce viruses. So say in viruses which come in the cell and force this machinery to produce their copies, but not the copy of the cell. Also they can reproduce themselves, but not anything. Yes, kind of, again, you don't know how to say. This suggests by far more interesting mathematics than phenomenon. Phenomenon is very kind of, it's rather primitive logic involved there. But in the logic in actual reproduction of cells, by far more interesting, yeah. And we don't have mathematical description of that. Really simple conceptual mathematics describing this process. And of course, everything happening in the random environment. Without all the steps, like for an enzyme, that depends on Brownian motion. Sometimes you might be hit by some molecule in order for something to happen, like diffusion. If you make synthesis of proteins, and the limiting step is diffusion of tRNA. So tRNA, molecule which carry a piece of RNA, triple sign on RNA representing this protein, this RNA, and the amino acid. They come together, and some of the part of the ribosome catches it when it comes the right one. And so it depends on the density distribution of particularly of these molecules. Yeah? So all this random noise crucial for functioning of the cell. At every moment, it makes selection. There is random kind of law, and then there are implementation of this law, and you take part of this implementation which is suitable. And this happens everywhere in life, on the molecular level and also on the level of evolution. Evolution by selection, random, perturbation and selection. And again, there is no mathematical formalism for that. We say these words, but what is selection mathematically? If you think about an intuitive word, what does it mean you select? Select what at all. It's systematic things happening in life every second, any millisecond in your cells. Right? It happens on the level of evolution. And everything what we do in life depends on this selection. But there is no mathematical, I believe there is no mathematical definition of what is selection, abstractly. Right? So my understanding, it is a distinction between, it's a kind of interplay between probability law and implementation of this law. And so, and this is two different probabilities. Okay, so I have to finish? No. You're willing to continue?
You have 20 minutes.
Huh?
20 minutes.
If it was 10:30, we should start at 11. Okay? Okay, so go to the next section. And this is, so this is about chicken and egg. Okay, now, so it is a, there are two other kind of aspects in biology where mathematics is kind of visible. So we see kind of, my point is that we see kind of traces of some interesting mathematics, but we don't know, it's not was actualized. But now the mathematics kind of entered very much with Darwin, and that's prior to that, as I said, was Bayes' discovery in biology, was Leeuwenhoek discovery of microscopic life, and really we do something completely new, which is people never could imagine beforehand, and with tremendously structurally organized entity, which was Buffon, and these people started to understand how complicated it is. But on the other hand, now some mathematics came, and this was Darwin, and in my view, he was 90% mathematics, a mathematician. His biology is negligible, and his contribution to biology kind of questionable, right? Most of what he was saying biologically was wrong, but mathematically he was right. He kind of emphasized that exponential growth of population and dying, most of them is an essential feature, which kind of perspective from which you have to look at evolution. Not that explain anything, but this perspective. And they question whether it was Darwin or somebody else, because, so immediately I want to bring some quotation. Yeah, so Darwin, but I was speaking about elephants, yeah. The kind of, computation of number of species for rabbits was done by Fibonacci about, 500 years before Darwin, and Darwin, kind of bigger man, that has done the same for elephants, and thus kind of the theory emerged, yeah, evolution theory, and he done it with mistakes. So very interestingly.
The mistakes. So what are the mistakes?
If you count, if you look at this number, you see you don't get these numbers. Yeah, you just arithmetic is wrong. Yeah, principle is okay. Yeah, but arithmetic is wrong. It's this interesting. I read some article also him about statistics. I forgot about some disease or whatever, and he also make mistakes in computation, but qualitative conclusion is right. So he kind of didn't care about for little things. And then, as you know, in his evolution picture, and everything was essentially wrong, but general direction was right. But the question is, all this funny kind of story about who was the first. So Boltzmann was very much excited by Darwin because he was explaining real life in a kind of mechanical, logical way. There is no purpose, it's just random something, and then being those which are, you survive, they are being fixed, and you go. But the question, how original was this idea? This is essentially all we can say about Darwin. Of course, he was looking how it happened in this case, reinterpreting everything. You replace God done, but it was done by God, and then, well, I said it was done by selection. And then nothing happens, it's just same logic. So it was really kind of looks, you replace everywhere God by natural selection, and you have new biology from old biology. It's no big deal. and the time was crucial because influence of religion was very strong, and the evolution for many was rejected on this basis. And there's an interesting example that even at the beginning of the 19th century, people believed that life can come from nothing, like from rotten meat. You may have flies, and the reason was that most parasitic, say, worms which live in the human body could have been created all the time, because otherwise, if we were from the beginning of life they were there, they would be all present in Adam, and Adam would have to carry all these parasites, and it was not really very happy life in Adam, having full with, all possible species of parasites. And it was really seriously discussed. Yeah? It was not kind of a joke. And Buffon, for example, couldn't say aloud many things he was saying, and in particular this guy Matthew, who wrote some book where he outlined evolution, he was not very much emphasizing that, probably also because at that time maybe it was possible to say, but not completely. And he was very skeptical about kind of originality of Darwin, and he certainly was believing he was discoverer of that. And there is this funny discussion in modern biologists. People, some say, well, Darwin was plagiarized and was not original, and there was Matthew, and there were some other people. And some say, well, Darwin, and that's it. And this is very also strange, as I said, biologists are very much ideological. They're very much emotional about that, yeah? Insisting that Darwin was so great and you cannot criticize him, or some, on the contrary, try to quite to say he was kind of plagiator, which is neither, I think, probably is true. And Darwin certainly was a great mind, but these ideas were floating around, and its originality is unclear who invented exactly what. And so what is interesting, even in modern time, you say there is Lamarckism, there is Darwinism, and they compare. But there is no difference at that point between Darwin and Lamarck. Both believe that that acquired traits are inherent. They go from generation to generation. With Weissman, who emphasized it's not so. And then, as we know, this idea was modified in the modern time, yeah, with epigenetics and things like that, and saying again Lamarck was not completely wrong. And the whole point, because even something happens in biology, you never know. It always happens, sometimes happens, it often happens, and so people argue they just based on this confusion, on this confusion with different concepts. What exactly Lamarck meant, but something being inherent or being produced in this way. And my understanding from where I read, when you firstly, that much about Lamarck people know come from what Cuvier said about him, but it was sarcastic. He wrote some sarcastic articles about Lamarck, personally making his statement look absurd. Yeah, because his kind of idea against evolution was that there is too much balance in our bodies, it couldn't come by small steps. And still we don't quite understand how it happened, yeah. Because there is very high correlation between our structure of our organism and how they're being formed. And it's in detail we still don't understand it. And secondly, when translate from French to English, because I think I forgot which word actually Lamarck used, saying that animal like giraffe taking some food, he stretches his neck, and this desire to tame something make effect on his body. And his completely logical idea that pressure, psychological pressure, not the movement, has effect on the body, which certainly may happen. And his idea was quite right. No, I mean, not absurd. It's not right, but not absurd. And then it was translated in English in some word. I'm not exactly how it was translated. So this was one of the confusion. And Darwin was always emphasizing that Lamarck has no effect on him, but he essentially kind of just lead some point going in his step, yeah. So it's unclear, of course, what it does come to what. But what actually mathematics there, and the point is that he suggested certain mathematical scheme describing evolution, and the scheme mathematically is as follows. Yeah, another point, of course, all this about this evolution, more or less what people, a big discovery in recent 200 years ago already was known to ancient, it was commonplace, I guess, yeah. According to Tite Lucretius, he was saying that. And so here is a model. So what we have, kind of mathematically, there is if you look in the point of view of Darwin, before it had any DNA, we have our organisms, and these organisms are characterized by certain traits. And for Darwin, these were numbers, were real numbers, like height, weight, taste, I don't know what. And there are many parameters, we don't know how many parameters, but probably they may be counted in million. Different parameters, and each of them may take value, say, from 1 to 10, continuously. And then you have the big cube, yeah? N is the number of traits, and this is we normalize them in the unit cube. And so what you care about point in this cube is the number of your traits given by numbers. At least, I don't think Darwin explicitly says it, but if you read him, he really means number because the Bible continues. And then inside of this cube, there is something, some possible traits which actually present and describe a particular organism, right? So we are in this language we describe with this vector, as they say, sequence of the traits, and this what you are, and then evolution means you move in this cube, and this is a picture of Darwin, and actually it's a kind of a random walk. You make some step, and some of them, and there is a death, dawn. If you go outside of some region in your life, you disappear, or you come back to the region when such trait give you viable organism. And then there are this probability of making step how the traits may change. And that's picture of Darwin. And this is the thing that he doesn't say it, but this is a picture. There is this dead, hugely world of dead things, tiny thing inside, which are alive, of these parameters, and you make random walk there. And we don't know what probability is, the steps are random, meaning, and so in what sense, by the way, are random? And again, so probability is kind of mathematical discipline, and there is some formalization. There are several, of course, models how you formalize it, but then how you apply it. And so how you interpret it. This is the most interesting thing, yeah? And of course, one of them, the key use of probability, is that if we have two things which are visibly independent, then the probabilistic independent, their probabilities multiply, right? And this multiplication rule of probability. But visibly independent, that you don't see relation between them, then you multiply them. And this is how you argue, right? So if you don't see connections, they multiply, right? Or you add some connection, you know they are, and everything else is independent. And of course, even I think Darwin realizes certainly not so, something may be correlated, right? And even we don't see relation, they may be secretly correlated. And this is kind of very essentially this is crucial. This kind of phenomenon is crucial in biology. Things are much more correlated than we may expect, but at least there is no purpose in that. This mutation may be correlated, but not because they know what will happen, right? But this only depends on the past. And so it's really a process where there is nothing teleological in this. However, in description of that, when you talk about that, you cannot avoid this point of view. This happens for some purpose. And Darwin himself, on one hand, he wanted exactly make it kind of scientific, so there is no purpose in this movement. But it's very difficult to avoid saying that, because there is selection, and selection has a purpose of survival, and the particular trait has some function, and so this function has a purpose, and so you do follow to some purpose. So there is reverse kind of causality there. So in natural science, everything must depend on the past, but here it depends on the future. You explain it, but what happens in the future with the purpose? And actually, Aristotle was describing the logic of causality. There are different causes, and some of the nature of things which produce them, and secondly, purpose why they were made for which purpose. And life has this feature, and so avoid it in the language is kind of hypocritical. It is there, and it is very impossible to speak otherwise, right? And this is an essential feature of describing when you think about anything in biology. You see why it is done in the sense of a purpose, which certainly never do in physics or chemistry, right? And but then anyway there is this thing, and there are some principles how you deal with that. So I just want to repeat, I wrote that. But the question, of course, we understand it very poorly. So we can look at many features of organism in their metabolism, which numerical features we can observe, how long they live and how many children they have and what are their size, hundreds of them. But the question is, it's subset. You can reduce, you can take some of these features, and then in the big cube they make a subset. But how does the subset look like? How big it is, and what is shape? And so you think that it's kind of on one hand, many parameters possible, some impossible, but you expect it to be something like Cantor type set, or this kind of fractal kind of set. We have some connectedness, some distribution, and it has definite dimensions, some Hausdorff dimension in there, and which corresponds to entropy essentially. So what it is? And we don't know that, and don't know really how to evaluate it, and I don't think it was ever anybody tried to evaluate it. But this is kind of essential, that when you make random mutation, still there is a chance you remain within the set, and so you have to know these probabilities, and we don't know them, and there is large body of mathematics kind of following this model, making lots of conjectures of actually what happened there. But they are conjectures. But this is a model of Darwin, and this is a kind of nice, this kind of life and death. And look kind of rather naive, but it tells you still something that the time you go from one point to another is proportional to the number of this trace, of these possible features, right? We have a cube, you kind of, the number of features, the time you go is proportional to the thickness of this cube. Of course, the number of elements in the cube is exponential, so choosing something there like that is completely unrealistic. And that's some reason why evolution is impossible, because the cube is too big, it's exponentially large, how you can choose it. But fortunately, going from point to point, it's only log of this size, still big. But it's kind of, if you make each mutation in each generation, you can see how many generations you have to go from one point to another, and this, you arrive at some numbers which, amazingly enough, very realistic, which Darwin found, but this was certainly basically related to understanding the geology, looking at different kind of, knowing the rate of creating different layers, geological layers, which actually was done before, 200 years before, by somebody else, but not so rigorously as done by Lyell and his kind of people. So this about Darwin, well. And so there is no explanatory power, of course, in that. It's just a way to describe things, yeah? And which, again, completely, in my view, completely kind of misstated by biologists. They say, oh, this explains something and give, of course, some satisfaction with some saying, but you can say it and you feel you understood it, unless, of course, you try to look at numerology of that and you see nothing is realistic. So it's explanatory power, very good for children. Yeah, but you can explain it, children understand this very well, but if you look, in actually contemporary of Darwin, we were very skeptical, and still we are skeptical that maybe in a point that we don't understand what happens. So of course, basic modification, as we know, are not that random and they're not small. And idea of Darwin everywhere, he was saying, it continues. He was emphasizing continuity, exactly how to make agreement of so many traits which must be correlated. And he believed you go by small step and can adjust them. But of course, the reason are more subtle than that. And we know the basic step of evolution of innovation is doubling of a gene. It's really a big thing. The whole gene doubles, and then it chooses either it may destroy you or it may take a new function eventually, but then by small adjustments. So there are at least two stages in that. And doubling is very common. Apparently, what somewhere I read, doubling of genome is very common, and most, say in people, this doubling happens very time, but then usually pregnancy being broken at a very early stage. Yeah, doubling of genome is deadly for animals, but it happens very often, and sometimes doubling of a gene or chromosomes, also not very healthy. But this is certainly a major fact. And then there are this lots of metaphors which confuse with reality, and which, on the other hand, need to be mathematically interpreted. Yeah, and this starting from concepts of a force. Yeah, so in my view, that many biologists, for them, force in physics and force like emotional force, they believe it's the same, or force of evolution, and they say very funny things. Even people apparently rather sophisticated. Yeah, actually there is somebody, person say Kaufman, who wrote many books about evolution, etc. And I believe he was a physicist because he had numbers. But sometimes, at some moment I read he was something incredibly idiotic about physics. Then I look at the internet, and he's a doctor by education. He was saying something about second law of Newton, obviously not understanding what it is. Yeah, and so he's a doctor, not a physicist. I thought he was a physicist because he loves numbers and everything. Yes, and now comes, after Darwin comes Mendel. Do we have time for Mendel?
Now we come.
We come to the end. Okay, so I just show one picture.
Selection.
Now we come to the lunch.
Yeah, so I want to show this picture. So now comes Mendel. This is very interesting to compare Darwin and Mendel mathematically and kind of conceptually. And again, I must say that most biologists always emphasize Darwin. From my point of view, as a mathematician, Darwin was just, synthesizer. Some say he was just compilator. On the hand, he was kind of great kind of synthesizer of ideas. And Mendel, somebody who produced new ideas, and he discovered new phenomenon. And this is the picture, and it has kind of the following corollary, which was perplexing biologists when they rediscovered Mendel at the turn of the last century, namely that if you have population, you have two kinds of flowers, and you mix them, and then you have this new distribution of color, right? So you had equal number of red and white. In the next generation, you have 3 times more red than white. And you look at the next generation again and think it will be 9-1. Etc. No, it stabilizes. No evolution. The fittest doesn't survive. The first generation, red look like fittest, and they kind of dominate, but then stabilizes. And they kind of, was very much unhappy, and just there's a story how somebody asked, playing some game with Hardy, somebody asked him, so what happened, and essentially he wrote this formula. Yeah. There is this equality. And of course, if you can say it in general words without anything, and it's completely, of course, obvious. But mathematically, it's obvious, and he just wrote some computation how you iterate the Mendel law, and this happens. But then it has this piece of mathematics, I think, quite amusing if you formulate it in these terms. Again, it's very simple transformation with quite non-trivial but still simple mathematics behind it. So here is a formula. You have a matrix, and you just make manipulation. You multiply, construct a new matrix, multiplying rows and columns. And the remarkable thing of this map, that's important. Square of this map equal to itself. And this kind of, in this term, is kind of elementary stuff. But if you think what behind it and what kind of geometric patterns, and it appears in many forms, just one thing I say, it's just Veronese map. This Mendel map in algebraic geometry is Veronese map, and there are Veronese varieties and lots of non-trivial mathematics attached to that. It's quite remarkable that, of course, you have polynomial map, and square of polynomial map becomes itself. Degree doesn't grow. It's very counterintuitive, both biologically and mathematically, and this interesting part of Mendel's law. So it's unlike highly intuitive Darwinian law wasn't accept this point of view. You turn a little bit your perspective, you see it. But here, discovery, you find it happens, and mathematically also it's a formula. You wouldn't expect it to be true. It is true, and it's many things actually. What interesting about that mathematically, this Veronese the map. So Veronese the map is you have you take space of linear form to space of quadratic form. Just take square of that, and then sphere goes to some surface, Veronese surface, in the high dimensional space. From dimension n, you go to n squared over 2, to space of quadratic form. And there are lots of geometric conjectures for this is counterexample. There are very many intuitive, well, several at least, very nice intuitive conjectures, very plausible, and counterexample found there. So it's counterintuitive mathematically indeed. This map is counterintuitive. And Mendel thing is counterintuitive, and this is discovery. So I'm just, I don't know how much Mendel understood, realized what he's done, and he was, by education, was a physicist. Right? He was, I forgot, he was major profession was physics. And, huh?
He was a monk.
He was a monk, but this because he couldn't find a job. But he was by education, he was actually worked with, I forgot the name of this, when you, how this speed of wave changes frequency. It is whose law? When something comes to you, it sounds Doppler. Doppler, yeah. Doppler was, I think he was professor. He was student of Doppler. And there was some other person, also a physicist, with whom he interacted. Yeah, he was, and he certainly knew mathematics and physics. So certainly much better than Darwin did. And this is a quite remarkable piece of mathematics, and indeed it is still the most mathematical part of biology around this transformation. And the various version of transformation with population, and this is actually this two is because there are these two parents. It's quadratic map. If there are more parents, will be higher degree maps, and there is a kind of quite rich mathematics there. And part of them is called Bernstein algebra, and then there are this I've forgotten this. They're called Jordan algebras, yeah? It's also algebras of this time. Some non-associative algebras was interesting thing, yeah. Okay. So, and then it was developed by so-called new kind of a new kind of evolutionary kind of development coming from Haldane and Fisher and Wright, who actually half mathematicians, and this is lots of mathematics there, which is, I must say, I don't understand how relevant it is to biology. It's still questionable. People argue it's very important, and many corollaries, but it's kind of semi-philosophical. But mathematics there is quite significant. Yeah, it's differential equation, very interesting differential equation, very interesting dynamics. Also appears in ecology, and chaos was discovered there by some of these people. I keep forgetting his name. In ecology, his name never come to me fast, if comes at all. No, it was not Lorentz. It was this guy. One second. No, I forgot his name. Yeah, there is an ecologist, this famous ecologist here, who physicist, who discovered this effect of this quadratic equation, who first discovered this chaos in the quadratic equation, with the quadratic iterations.
You mean Mandelbrot?
No, it was rather recent. He was in ecology. He did it for ecology. Not West, but... Yeah, anyway, I forgot his name. I just definitely have excuse, yeah, for forgetting names. But there are lots of mathematics there, but it's the moment mathematics starts, I think biology a little bit stops, yeah, right? That don't fit well. Okay, yeah. So now it will be 10:30. I understand, yeah. All other days, oof.